Inhibition of SARS-CoV-3-like protease activity by theaflavin 3,3 digallate, TF3. By Qianan Chen 1 Kony PC Lin 1 Kuo Ki Huang 1 Wei Cheng Chen 1 HSIN Peng Si 1 Po Huang Liang 2 and John Ta. HSU 1,3. SARS-CoV is the causative agent of severe acute respiratory syndrome, SARS. The virally encoded 3C-like protease, 3CL-PRO, has been presumed critical for the viral replication of SARS-CoV in infected host cells. In this study, we screened a natural product library consisting of 720 compounds for inhibitory activity against 3CL-PRO. 2 compounds in the library were found to be inhibitive, tannic acid, IC50 equals 3M, and 3-isotheaflavin-3-gallate, TF2B, IC50 equals 7M. These 2 compounds belong to a group of natural polyphenols found in tea. We further investigated the 3CL-PRO inhibitory activity of extracts from several different types of teas, including green tea, oolong tea, Puer tea, and black tea. Our results indicated that extracts from Puer and black tea were more potent than that from green or oolong teas in their inhibitory activities against 3CL-PRO. Several other known compositions in teas were also evaluated for their activities in inhibiting 3CL-PRO. We found that caffeine, epigallocatechin gallte, EGCg, epicatechin, EC, theophylline, TP, catechin, C, epicatechin gallate, ECG, and epigallocatechin, EGC, did not inhibit 3CL-PRO activity. Only theaflavin 3,3 digallate, TF3, was found to be a 3CL-PRO inhibitor. This study has resulted in the identification of new compounds that are effective 3CL-PRO inhibitors. Introduction. Severe acute respiratory syndrome, SARS, is caused by a newly discovered coronavirus, SARS coronavirus, SARS-CoV, 1,2. Like other related coronaviruses, SARS-CoV is an enveloped virus containing single-stranded RNA of positive polarity. Coronaviruses have the largest genomes known, 30 kb, among RNA viruses. The genome of SARS-CoV comprises one open reading frame, ORF, encoding 2 overlapping replicase polyproteins, polyprotein 1a, pp1a, 450-kta, and polyprotein 1ab, pp1ab, 750-kta, responsible for virus replication. The rest of the genome comprises at least 5 ORFs encoding structural proteins and other genes with putative functions. Proteolytic processing, especially the processing of replicase polyproteins, is one of the crucial steps in the life cycle of many positive-stranded RNA viruses, including coronaviruses. All coronaviruses encode a paplin-like protease, PLP, and a chymotrypsin-like, 3CL-PRO, protease for proteolytic procession during virus maturation. Studies on other coronaviruses have shown that the PLP protein cleaves at no less than 2 sites on the pp1a polyprotein and that the 3CL-PRO protease cleaves at least 11 interdomain sites on the pp1a and pp1ab polyproteins. Thus, the 3CL-PRO protease of SARS-CoV has been considered as an important molecular target for anti-SARS-CoV drug discovery and developments. In this study, after screening of a natural product library and further confirmation, we found that SARS-3CL-PRO could be inhibited by compounds that are abundant in teas. We also examined crude extracts from various teas and a panel of representative natural products in teas for their inhibitory activities against SARS-3CL-PRO. Methods. Preparation of SARS-CoV-3CL-PRO. Total RNA extracted from a throat swab of a SARS patient was provided by the Center for Disease Control, CDC, Taipei, Taiwan. All RNA samples were handled under Biosafety Level 2, BSL-2, regulations. 2 primers, 5-JOT-TACH-KAKA-3 and 5-JOT-KA-IJKA-KOTGATGIJTC3, were used for reverse transcription polymerase chain reaction, RT-PCR, to synthesize the cDNA of SARS-3CL-PRO using ready-to-go RT-PCR beads, Amersham Biosciences, Piscataway, New Jersey, according to the manufacturer's protocol. The PCR cycle used for the amplification of cDNA of SARS-3CL Pro was as follows: 94 degrees Celsius for 5 minutes, 25 cycles of 94 degrees Celsius for 1 minute, 58 degrees Celsius for 3 minutes, 72 degrees Celsius for 2 minutes and 72 degrees Celsius for 10 minutes. A full-length 3CL PRO of SARS coronavirus was successfully expressed in Escherichia coli as a glutathione S-transferase, GST, fusion protein. Following cleavage by thrombin, 3CL PRO was purified to homogeneity. A 1,5-MER peptide with sequence H2N sitsafulxkferkama COOH corresponding to an autocleavage site of the SARS-3CL Pro was synthesized and used for protease cleavage activity assay. The highly purified protein was shown by high-performance liquid chromatography, HPLC, analysis to exhibit proteolytic activity in trans. Preparation of crude extracts from oolong tea, green tea, black tea, and Puer tea. Tea liquors were prepared from oolong tea, green tea, black tea, and Puer tea, also called Puer or Puer. Samples as 2%, W/V, tea solutions. The water extracts of 4 different types of tea leaves were prepared by shaking for 10 minutes in boiling hot water in thermal flasks. The extracts were then filtered through a Millexgs 0.22m filter, Millipore, Malsheim, France, to remove particulate matter. The water extracts were then evaporated under reduced pressure to obtain viscous masses. These materials were evaluated for their inhibitory activities against SARS-3CL Pro. Inhibition of protease activity of SARS-3CL Pro by HPLC assay. A general cysteine protease inhibitor, N-ethylmelamide, NEM, protease inhibitor as positive control, was obtained from Sigma, St. Louis, MO. This compound was tested for its inhibitory activities against SARS-3CL Pro in the peptide cleavage assay. The activities of the proteases were measured in the absence or presence of different concentrations of these general inhibitors. The enzyme, 2M, was pre-incubated with chemicals for 30 minutes. Peptide was then added at 100 m to the reaction, and mixtures were incubated at 37 degrees Celsius for 1H. The potency of the inhibitors was calculated. The HPLC analysis condition is, a C18RP guard column, 250 x 4.6 mm x 5 m, Agilent Zorbax extend. This method was chosen because it gave the best resolution among several methods that were tested. The column temperature was ambient, and the elution, 0.8 ml/min, was performed using a solvent system comprising solvents A 10 mm NH4OAC, 0.1% trifluoroacetic acid, TFA, and B, acetonitrile, 0.1% TFA, mixed using a gradient starting with 100% A, linearly decreasing to 10% A in 8 minutes, to 100% A in 16 minutes, and held at 100% A for 23 minutes. The injection volume was 40 ml. The UV detector was set at 214 nm. Results: evaluation of tea extracts and pure components from teas for their inhibition of 3CL PRO activity. Since many other polyphenols related to tannic acid and TF2B are also present in various kinds of teas, we decided to examine the inhibition of activity by various tea extracts and several well-known pure ingredients present in teas. Thus, water extracts from different types of teas were prepared and evaluated for their inhibitory activities against 3CL PRO. As shown in Table 1, extracts from black and Puer teas inhibited 3CL PRO activity while those from green or oolong teas did not. We further tested the possible involvement of several known ingredients present in teas, including caffeine, theophylline, catechin, C, epigallocatechin, EGC. Epigallocatechin gallte, EGCg, epicatechin, EC, epicatechin gallate, ECg, TF1, TF2, TF2b, tannic acid and TF3. The results showed that methylxanthine, caffeine and theophylline did not influence 3CL PRO activity. Catechins, including C, EC, ECG, EGC and EGCg, present in green tea, unfermented, and oolong tea, partial fermented, also did not inhibit 3-CL-PRO activity. Our results indicated that tannic acid, TF2B and TF3 are 3-CL-PRO inhibitors as revealed by the fluorogenic substrate assay. TF2A was not tested because of its unavailability. Discussion. Human coronaviruses, CoVs, are major causes of upper respiratory tract illness in animals including humans. It has been demonstrated that a novel coronavirus causes SARS. The main protease, frequently called 3C-like protease, 3-CL-PRO, has been identified as an attractive drug target. Of the 720 natural products screened in this study, tannic acid and TF2B were identified using the HPLC proteolytic assay to inhibit 50% of the proteolytic activity of 3-CL-PRO at concentrations less than or equal to 10 m. Both tannic acid and TF2B belong to the group of natural polyphenols found in teas. Low concentrations of tannic acid were found to inhibit proteases including tissue-type plasminogen activator, urokinase-type plasminogen activator and plasmin activity. Over 300 different kinds of tea are now produced, but they are classified into three main forms. Green tea is manufactured by drying fresh tea leaves, with emphasis on the prevention of oxidation of the tea polyphenols during the manufacturing process. In contrast, the manufacture of black tea is characterized by a high degree of fermentation, which produces a series of chemical condensations among the ingredients present in tea. Oolong tea is partially fermented, and the tea compositions are more similar to those found in green tea. In evaluating different types of tea, we found that extracts from several different types of tea, including Puer and black tea, could inhibit 3-CL-PRO activity while green or oolong tea could not. However, the chemical composition in Puer tea is very complex and it is difficult to isolate pure ingredients for structural identification. Purification and identification of Puer tea ingredients should be carried out in a separate study. We next examined whether other well-known ingredients present in tea could also inhibit 3-CL-PRO activity. We found that methylxanthine, caffeine and theophylline, and catechins, EGCg, EC, ECG, and EGC, were not able to inhibit 3-CL-PRO activity at concentrations up to 100 m. However, TF1, TF2 and TF3 were more potent 3-CL-PRO inhibitors than catechins in green tea. During the fermentation step in the production of black tea, most of the catechins are oxidized and condensed into theaflavins through dimerization and into theophylines through polymerization. Green tea contains 30% of catechins, dry mass base, while black tea contains 9% of catechins and 4% of theaflavins. In another study, Long et al. 16, reported that TF3 was the most abundant, 1.05%, theaflavin in black tea followed by TF2A, 0.34%, TF2B, 0.11%, and TF1, 0.08%. Tannic acid is a class of polyphenolic in plants, but the quantification of its levels in green or black tea is difficult because tannic acid constitutes a wide range of polymers with mol. WTS of 503,000 day. It is interesting to note that TF2B and TF3 are more potent inhibitors of 3-CL-PRO than TF1. Unlike TF2B and TF3, TF1 does not contain a gallate group. Thus, the addition of a gallate group attached at the 39 position to TF2B and TF3 might be important for their inhibitory activity against 3-CL-PRO. These results suggest that TF2B and TF3 might be good starting points for the design of more active inhibitors for the 3-CL-PRO encoded by SARS-CoV. Finally, this study has identified three compounds, TF2B, TF3 and tannic acid, that are effective 3-CL-PRO inhibitors, IC50 less than or equal to 10 m. These compounds are abundant in the extract of black tea. Black tea is a popular beverage in the world. Results from this study warrant further investigation to examine the effect of these natural products in inhibition of SARS-CoV replication in cell culture. Clark et al. Reported that theaflavins extracted from black tea were able to neutralize bovine coronavirus and rotavirus infections. Thus, it will be very interesting to evaluate, in a separate study, whether drinking black tea can prevent or alleviate the infection of an enteric form of coronavirus, since SARS-CoV is known to actively replicate in the intestinal tract. Acknowledgements. We thank Dr. Tsongder Wei for providing excellent technical support. This work was supported in part by funds and facilities of NHRI of Taiwan and by grants NSC 92-2751B 400-006Y to XC and JTAH and NSC 92-2751B 001-012Y to PHL from the National Science Council, Taiwan.